Our next speaker is Dr. Kim Goodspeed, who I introduced yesterday, so I don't think she needs another introduction. She's going to be talking to us about GTX-102, what we've learned together. Kim.
Hello, everybody. Thanks so much for the opportunity to be here today again. For those of you I didn't get to meet yesterday, I'm Kim Goodspeed. I'm a child neurologist by training, been with Ultragenyx for a bit now, and I get to have the distinct honor and pleasure of being the clinical development lead for our GTX-102 program. And of course, GTX-102 is our antisense oligonucleotide ASO for Angelman syndrome. Before we dive too deeply, though, I did want to say thank you again. So thank you to FAST for letting us come be part of the programming here this summit. It's been an amazing summit, lots of exciting science happening. Thank you to those of you out there listening and interested to learn more and dive into stuff that's really complicated. We really appreciate all of your questions and your feedback. Thanks to our participants and families who have trusted us to go on the journey with us and been part of our trials to date. And to those study teams out there who are making all of the magic happen at the study sites so that we can actually do the studies, answer the questions, and move things forward. So thank you, everybody. So disclaimers, of course. GTX-102 is an investigational drug, and I'm presenting open-label interim data. So we're going to share some data from our phase 1/2 study. You've seen this before, but we're thinking about it in a slightly different way and getting to dig deeper into the methodology. For those of you who were here yesterday, some of the slides are repeated, but my grandmother was an elementary school teacher, and I think I learned from her that it's really important to hammer home messages and repeat things that are important. So we're going to go through those slides again. And of course, I'm an employee of Ultragenyx. So who's Ultragenyx? All we do is rare. We do rare and ultra-rare diseases, and we were founded in 2010 by Emil Kakis, and he saw a problem. He was working on MPS 1, mucopolysaccharidosis type 1 at the time, and saw that it was really hard to do drug development for rare diseases, and he thought he could make a difference. So he's now built out a team of 1,300 employees. We're based out of Nevada in Northern California, but I live in Texas. We've got people all over the globe that are working to help bring new drugs to disorders that don't have treatments right now. And this is GTX-102. So we'll just spend a tiny bit of time here. I think this audience knows what ASOs are by this point, but GTX-102's job is to bind to the antisense transcript and allow for expression of the paternal allele. So it does that by binding to that transcript, causing it to be degraded. That opens up the paternal allele for expression so that you get to use that backup copy from dad to make UBE3A protein. We have looked at a lot of different outcome measures and see that we're seeing some improvements across some of the core symptoms of Angelman syndrome, and we've had a generally pretty good safety profile. So here's some of the most common safety events that we've seen in our phase 1/2 trial. We do have a safety event that we monitor closely. It's an adverse event of special interest, or AESI, or you may have heard the term radiculopathy in some of our other presentations. What that essentially means is that we've had a few kids who have had some sensory changes or some motor changes that have impacted how they were able to walk for a short period of time. They all recovered completely, nobody had any long-lasting impact, and were able to go back on drug and have improvements without having any recurrence of this event. So it's an important event, it's an event that we monitor for, but we know how to monitor for it, we know how to identify it, we know how to treat it, and most importantly, we know how to get kids back on drugs so that they can continue to receive potential benefits from GTX-102. So the way we've structured this talk today is we're in a nice, interesting time in our program where our phase 3 study is running right now, and so we've got a little bit of time to reflect, a tiny bit of time, I think, but we were thinking about all of the lessons we've learned from you as a community and how much you've made our program better. So lesson number 1 was measure what matters, and so y'all are the experts on Angelman. You know what it's like to live in your household with your loved ones that have Angelman syndrome and the daily challenges that you face. And it's our job and my team's job to figure out how do we turn those daily challenges into things we can measure as clinical outcome measures. So one of the ways we've done that is by learning from your natural history studies, learning from your disease concept studies, and then also learning from our own data. So we can look at our baseline data, and we've got a poster up that summarizes all of this. But what I thought was really exciting and our team was excited about was that this data really reflects that we're able to measure and quantify the symptoms that you've told us matter. And what that means is that we're able to put a number to it, and that's a number that we can follow over the course of a clinical trial and see if it changes. And so these data highlight the importance of capturing those symptoms. We capture them in a quantifiable way through those surveys, the Bailey data, Vineland data, all of these things, but we also get to have qualitative data from our caregivers that are participating in the trial. So we do qualitative interviews at study entry and periodically throughout our studies with a subset of participants, subset of caregivers, and they tell us what all of this stuff means. What is it like to be in the trial? What is it like to live with your loved one that has Angelman syndrome? What is it like when you say behavior problem, what does that mean? And then we can link that data with our quantitative data and give it some more color and meaning. But one of the ways we also like to look at our data is with the multi-domain responder index. So again, we've got a cool poster that I'll flag and you can go talk to us about it. And we learned from the PFDD, excuse me, we learned from the PFDD that sleep and behavior are also super important in addition to cognition and communication and seizures and all of the other things you face. And so we got to collaborate with FAST and were asked to look at our behavior data, and it was a really cool opportunity to say, look, our behavior data is capturing the symptoms of hyperactivity, noncompliance, impulsivity that are really challenging in the home, and we see it changing in response to GTX-102. So now we know that we've got some pieces that we get to go after, look at even more depth in our phase 3 study. But that's just one piece of the puzzle, right? And so the way that we like to look at our data is in all of the symptoms at one time and see how things change. And that's because a single primary efficacy endpoint may not be good enough to measure all of the ways these drugs can impact patients. The way that drugs are typically designed in clinical trials to be tested is you pick one symptom that's linked with one clinical outcome assessment, and you need to see change in that one particular measure. That means that everybody that enters the trial probably needs to have that symptom. For some disorders, that may be okay. For some symptoms, that may be okay. But what if you're missing other potential benefits? Or what if you're having to exclude participants who could benefit in other ways, and they're not getting to be in that trial, and we don't get to learn about their experience too in other symptoms? We also know that many drugs impact patients differently. One drug may treat two different symptoms, and isn't it important to know about that and be able to, put significant weight behind that? And for that matter, we've heard all the exciting stuff about the ASOs, the CRISPRs, the gene therapies this morning. These are all targeting the underlying symptom or the underlying cause of Angelman syndrome. And we know that your kids are not all the same. Yes, there's a group of symptoms that we're expecting to see, but we don't see every single symptom in every single person in the exact same way. So the multi-domain responder index, or the MDRI, is one of our solutions to be able to capture and assess this change across multiple symptoms using one analytical tool. So what is it? It's a statistical analytical tool. It's a methodology that we apply to our data. So we assign clinical domains that are meaningful based on the patient population we're studying, and then we assign clinical thresholds to those and see what our responder analysis looks like. We'll go into that in a little more detail in a second. If you've heard of composite endpoints, that is another way to pull in multiple clinical endpoints into one analytical tool, but it's a little bit different. In the MDRI, each of the domains is independent. We're measuring it individually, and in this case, they're all symptoms of Angelman syndrome. But for a composite endpoint, you typically need it to be a single symptom that you're measuring in different ways, and all of those outcome measures are pointing to that one symptom. So different ways to measure one versus different symptoms being measured at the same time. So we do it by identifying 4 to 6 different clinically important domains. They need to be important and independent. Then we assign a clinical endpoint to each of those domains or each of those symptoms, a way to measure for change. Then we define the meaningful score difference. It used to be called a minimal important difference. There's been a lot of different terms for this. What it's essentially saying is a clinical threshold. Beyond this score is good, below or beyond this score in that direction is bad. So if you meet or surpass the clinically meaningful threshold in the direction of improvement for the scale, because remember, some scales go up and some scales go down for positive or negative. If you meet or surpass in the direction of improvement, that means that we give you a green box and you're a +1, and we say you had improvement. Meet or surpass in the direction of worsening, we give it a pink box, you get a -1 point, and we say that was a case that worsened. If it falls somewhere in the middle in the gray zone, we say that's an unclear treatment effect. We leave the box white and we give it a score of 0. You tally up the scores for each participant. So in this hypothetical dummy data here, you've got 2 domains that improve, 1 domain that worsened, 2 domains that were unclear. You get a net response of +1. Then we apply statistical methodologies to that, statistical testing to the group, and understand if we have statistically significant changes or not. So, how did we build an MDRI for Angelman syndrome? We started with our domains. Again, we learned these from you. The disease concept studies, the natural history studies tell us that these are the symptoms that don't have good clinical medical treatment options right now. We assigned a clinical endpoint to each of these domains. So, based on our phase 1-2 data, we're able to learn, okay, we can measure sleep in a bunch of different ways, but which one is the best? Which one do we think is capturing what the patient community tells us is important? And which one is also responding to treatment? So we see change scores over time. And so that's how we picked each of these endpoints, was based on our phase 1-2 data. Then we get to define the MSDs, so the meaningful score difference for each of these. And again, we use our phase 1-2 data to help us understand what is the threshold that we need to say, if you're beyond this score, it's an improvement, if you're beyond this score, it's a worsening. So let's walk through a couple of examples, and then I'll show you the entire table. So first, a relatively simple one. So we have participant 1 here. They got better in every single one of these domains. So they met or surpassed in the direction of clinical improvement on each domain. That means we color the box green and we give it a +1. So they have a total net response score of +5. That's the max score we could get on this. Subject 2, which is technically subject 11, our second example. So this one's a little bit more complicated. We've got 3 domains that improve, 2 that stayed essentially the same, had an unclear treatment effect. So they get a net response of +3. The last one, so this one has 2 domains that worsened this time. So we color those boxes pink, so they're a -1 point. 2 boxes are green, that means they surpassed the threshold in the improvement direction, so they get +1 for each of those. And they had 1 that was an unclear treatment effect. Add those all up and you get a net score of 0, okay? So that's how we walk through each participant. Now the whole big data. So this is all of our participants from cohorts A and B, so just 2 of our groups that were in our phase 1-2 study. We're assessing them at baseline and at day 338, which is approximately 1 year into treatment, and we see how did their score change over the course of that 1 year. Did the score meet or surpass the score differential for the direction in improvement or for worsening? And so if it's improved, we get a green box, worsened pink box, no change, white box. Add them all up for each participant, and then you get that final column there, which is the total net response. And you can see the total net responses in green tally up to 22 out of 28 of these kids, which is 79% of kids with at least 1 domain that has a net responsive clinical improvement. You can see that some kids improve in some places, don't change in some places, and even worsen in some places, but it gives you a sense that everybody's responding in different ways, and we get to look at that data in this kind of bird's eye view of the totality all at one time. Now, of course, we look at each of these individually as well, in the more traditional way of looking at our line graphs and our bar graphs that we show you all the time, but we think that this is a really nice snapshot that gives you a sense that a lot of kids are improving. Okay, so lesson 2, patient-focused study design. I think one of our core tenets at Ultragenyx is that we do patient-focused drug development. We get your input all along the way, and in fact, you have given us a lot of feedback and input over time. So we have more formal, less formal interactions with you, and we appreciate that feedback always. We do our best to hear you, to listen, and to make changes to the best of our abilities. So thank you for the feedback, and know that we'll continue to keep asking. One of the ways that I'd like to highlight that we did receive feedback and made some changes was how we thought about our phase 3 study. So to walk you through a little bit, how do you design a controlled clinical study? Traditional way is a placebo-controlled study. So you have a placebo group that receives a medicine that is essentially like a fake medicine. It's a medicine that looks and is administered and given exactly like the treatment drug with active treatment, but the placebo doesn't have any active ingredients in it. In rare disease, it's tricky though. We have to be creative, right? We have small patient populations. Your kids are also vulnerable, and we need to protect them and think about what do we absolutely need to do to answer our scientific questions. We also had feedback from the EMA that came from those of you in the audience who are our European colleagues and families. You got to give some feedback to the EMA, and they summarized that in a survey, and we heard you loud and clear that sham-controlled was preferred. So what's a sham study? So a sham control means that you don't actually give any medicine or fake medicine. It means you do a procedure and you make it look like you're giving the medicine without that final step of administering anything. So that's the study design that we carried into Aspire, our phase 3 clinical study, which of course we got feedback from you on. So our phase 3 study, Aspire, is our global study. We got to enroll 129 kids, and kudos to you in the audience and all of our investigators that worked with us on this. We enrolled this study very quickly. And by July this summer, we were completed for enrollment, and now that data just gets to cook like a souffle. We're not touching it until it's ready to be looked at. And so it's studying the same population that we studied in our phase 1-2 study, patients 4 to 17, deletion genotype, and we're looking at the same similar outcome measures that we looked at there. It's a sham-controlled design again, but it is double-blind. So that means that everybody goes through a screening and randomization period, and then you're randomly assigned 1-to-1, so a 50-50 chance to either get treatment in the double-blind period or to go into that sham control where you are getting a sham procedure completed at every visit. And for some of those visits, we're doing a full lumbar puncture to pull off a little bit of spinal fluid so we can do some analyses, understand some more safety profiles, and also continue to work on biomarkers because they're so important. So the first year of the study is double-blind. That means that most of the study site, or a group at the study site, are blinded and don't know whether the participant is getting drug or is getting the sham. The caregivers don't know, the participants don't know. There are some study site staff that do have to know because, right, something different happens in the procedure room for each of these kids. And so they need to know if they're giving drug or if they're doing the sham. But in the open label period in year 2, everybody receives drug. So the treatment period, the treatment arm gets to just continue receiving their drug in the maintenance period. The sham control arm will transition over to drug in that open label year and go through the same dosing regimen as all of the other participants have. Okay, lesson 3, inclusion of all genotypes is important. We've heard you multiple times. We also agree GTX-102 was designed to treat Angelman syndrome. It was designed to restore UBE3A production regardless of the underlying genotype, regardless of age. And so we need to look at the safety and efficacy in these other populations that we haven't had the opportunity to study yet. So as a broad overview of clinical trials and the phases and how they kind of work, this isn't meant to be a linear timeline, but you march through typically phase 1, phase 2, and phase 3, and they each have different goals. So your phase 1 and phase 2 studies are your safety studies. In rare disease, we always look at efficacy too, but they're your safety studies. That study completed enrollment quite some time ago at this point, and so those kids are continuing to receive drug in our long-term extension study, and we get to continue to learn about the long-term effects of GTX-102. Phase 3 studies are your pivotal, your controlled studies that are really meant to truly assess benefit risk. Is it working and is it safe? And you typically control them through that blinding mechanism like we just talked about. Enrollment completed for this study over the summer. We're just waiting for the data now. But we're here now. So phase 2 studies, again, we get to do safety studies. Remember, we need to look at the safety and efficacy in our other populations. And so this study actually opened for enrollment in October, and we're really excited. If you want more information, thank you. Thank you. We're super excited. We're very proud of the team. Appreciative again to the investigators who go on this journey with us and go really fast. We are so excited to have this study going and can't wait to see what this data looks like. So looking at them one more time, phase 1/2 study was our safety study. It was the initial study, kids ages 4 to 17, deletion genotype, really helped us design the rest of the studies going forward. Our long-term extension is where we get to see that long-term benefit, the long-term safety profile when kids are on maintenance therapy. And this is the study where kids will always land from their parent study, so from the initial study they enroll in. Phase 3 is the Aspire study. It's our pivotal, well-controlled study, and it finished enrollment this summer. And Aurora is our phase 2 study, look at safety and efficacy in the additional populations we haven't gotten to study yet. And all of this information can be found in much greater detail at ct.gov. So I encourage everybody to get familiar with that website if you haven't already, and just try to digest the government website as best you can. So what is Aurora? It is a global study. We're enrolling approximately 60 children and adults, and it's going to be a phase 2 basket design study. That means that we get to include different subgroups and study them under one clinical protocol. We're looking at all the same kinds of symptoms, but it may be slightly different to look at behavior in adults versus behavior in younger kids. And so we've thought through all of those details and tried to pick the right outcome measures to measure these symptoms in each of these populations. So group A is going to be deletion genotype, and these kids will be ages 1 to 3. Group B is going to be our UPD and ICD kids, and they're going to be the same ages, 4 to 17, as our other study populations. Group C is our adult group, and we're kind of being more flexible with this group and accepting any genotype into it. And then group D is our mutation type genotype group, and those again are 4 to 17 years old. And so our groups A, B, and C are going to do a straight open label 1-year study design. So it is open label, meaning everybody's on drug and everybody knows everybody's on drug. You go through your screening and randomization, and then they get treated. We assess safety, we assess efficacy endpoints along the way, and then eventually they get to roll in the long-term extension study. And then group D, because we have learned that there can be a little bit more variability in the developmental severity of this group. We understand that neurodevelopmental disabilities is ubiquitous. Everybody with Angelman syndrome has significant neurodevelopmental disability, but this group is the group that has just a slightly bigger variation in that level of severity between individuals. So for that reason, we've included a no treatment control period in this study. So what will happen is kids will every 2 kids that go into the treatment open label 1-year arm, 1 child will go into that no treatment control period and then start treatment after about 6 months. So it's a 2-to-1 randomization. It's still open label, it's still not blinded, it's still not sham, it's not placebo. Everybody will get treatment. We're just asking for a 6-month run-in period for 1/3 of these kids so that we have 2 different time points before treatment. That helps us understand a little bit better what they looked like before treatment, and then we can see what they look like after treatment. And we can also still compare them to those that go straight into treatment. So it just gives us even a little bit more information. I understand it's a bummer to be in a no treatment control period for 6 months, but it really will help us answer the question scientifically, I think, in a more rigorous way. And as I mentioned, everybody gets to roll into that long-term extension study should they choose to continue on the journey with us. So drug remains available to patients after their participation in Aurora. So how can you get more information? We have a team out here, of course, and we also have a team that you can email directly. They're able to take your information, help connect you to clinical trial sites that are close to you, help you answer some questions even before you get to the site, help you think through what questions might be good to ask. But as Allison mentioned earlier, please talk to your doctors, talk to your families, talk to other families and each other here to understand what it's like to be in a clinical trial and decide if these are the decisions you want to make for you and your family. So I will end here. Our phase 1/2 trial showed promising benefit-risk profile, meaning we get to continue to study. Our global phase 3 Aspire trial completed enrollment this summer, and we get to look at that data when it's ready. All of our participants continue to receive drug through our long-term extension study, and we get to understand those long-term impacts from safety and efficacy both. And then Aurora has opened. We're super excited, and you can get more information on that study at ct.gov. And then finally, the MDRI we think is a powerful tool, so one last plug for it. We think we can measure meaningful improvement across multiple domains at one time, and we think that's a really nice way to look at efficacy data in a complex disorder like Angelman syndrome. So thank you so much.
Incredible, and thank you for all of that. And that's a whirlwind of an unbelievable amount of work over the last 5 years. And so it's amazing where we are and how far we've come. And I just want to acknowledge to the community that the day the press release came out that the first patient was dosed in Aurora, I got an email from Emil Kakis that said, "I told you we would keep our promise." And that's because we made them promise, as we have to all sponsors, that they would include all ages and all genotypes, and they did.